Have you ever wondered why cephalopods such as octopi and squids can change color to match their surroundings and hide from predators? For a long time, their techniques remained a mystery. However, the mechanism used by these creatures has finally been discovered. Proteins that enable these cephalopods to change color are called reflectins. These reflectins control the reflective pigment cells called iridocytes. Chlorophyll, found in the leaves of green plants, is an example of a pigment. It absorbs light from the red and blue regions of the spectrum and reflects green light, causing us to perceive leaves as green. Unlike chlorophyll, iridocytes use transparent guanine crystals to reflect the light of certain wavelengths. The cephalopod's nerve cells release the neurotransmitter acetylcholine to trigger a cascade that eventually leads to the phosphorylation of reflectin. A single reflectin protein is like a string of beads. They reside in finger-like protrusions of membrane called lamella. Since reflectins are positively charged, they repel each other, causing the protein to straighten. However, the addition of negatively charged phosphate groups neutralizes the positive charges, causing the protein to fold, which exposes hydrophobic regions. Each reflectin can connect with up to 4 phosphates, and the more phosphates, the more folding. Because hydrophobic objects repel water, the folded reflectins clump together. This causes the fluid pressure to change and pushes water out of the lamellae, reducing its thickness. This causes a shift in the color of reflected light. The thinner the lamellae, the closer the reflected color gets to the wavelength of blue light. The removal of the phosphate groups causes the clumps to revert back to individual reflectin molecules and the reflected color to shift back to transparent as the water flows back in. This discovery not only explains the cephalopod's color-changing abilities, but can also provide advancements in other fields. The mechanism reflectins use is similar to the proteins involved in degenerative diseases such as Alzheimer's. Further studies may unveil a way to reverse the pathological effects of these diseases.